Good morning, and welcome to the second of two videos on the second section of Chapter 3, How We See and Hear. You'll recall from the first video, which hopefully you watched just a few moments ago, that we talked about the anatomy of the eye. In this video, we're going to talk about theories of how the light that is sensed is actually perceived by the nervous system and specifically the brain. Now, in terms of sight, there are three major theories about how this actually works. And the first of these three is what we call the trichromatic theory. And this is a theory of color vision that assumes that there are three types of cones, each only activated by wavelength ranges of light that correspond roughly to blue light, green light, and red light. Now, you'll remember that the wavelength of light determines what its color actually is. So in terms of, for example, blue light, you've got a very short wavelength of about 400 nanometers. For red light, you've got a very long wavelength of light of about 700 nanometers. And green falls right in the middle, right? The Roy G. Biv from the colors of the rainbow. And so certain wavelengths of light stimulate certain cones, right? The red cones, the green cones, the blue cones. So how does that make colors other than blue, green, and red? All right, well, the trichromatic theory assumes that all of the other colors are simply mixtures of red, blue, and green. And so certain cones are stimulated at various levels of activation. And what that does is, you mix these colors together and you get a third color. So we call the blue, red, and green the primary colors and all of the others secondary colors. So if you want orange or yellow or violet, those are a mixture of blue, green, and red. And of course, if all three of these cones are activated at the same time and the same level of intensity, we see white light. Okay, because all of the colors mixed together give us white. Now, this is what's known as the additive mixture idea. And that's illustrated in the diagram on the right-hand side, all right? Notice that if you put green and red light together, you get yellow. If you put blue and red, you get magenta. And if you put green and blue together, you get cyan. Now, magenta and cyan are not colors of the rainbow. Those are just different shades. But notice what happens right in the middle, right here, all right? We get white light. Okay, and so red and green make yellow, red and blue make magenta, green and blue make cyan, and red and blue and green together make white. All right, what do they call this? A Venn diagram, I think. However, interestingly enough, while this might work in our eyes, and it certainly works in, for example, television technology. If any of you have a very old television, a projection TV screen, you'll note that there are three light bulbs in it. One is red, one is blue, and one is green. All right, and that makes all of the various colors on the TV screen and in your eyes. But if you were to take three paint cans, a can of green paint, red paint, and finally a can of blue paint, and mix them all together, guess what? You get black. All right. And the same thing is true if you mixed yellow, magenta, and cyan. Right in the middle here, you get black. And so in certain scenarios, like with paint, not necessarily with light, what happens here is that the mixture of the wavelengths, some of the wavelengths get absorbed instead of reflected, and therefore they are subtracted from the mix, and so they don't get reflected to your retina. All right. So red, green, and blue light together make white light, but red, green, and blue paint together make black paint. Okay. And then, of course, the additive mixtures idea doesn't lend itself to producing white light in certain complements. For example, if you take red and green paint, light, and put them together, you should get white, and blue and yellow should give you white, but it doesn't always necessarily work that way, at least in terms of mixing other things. All right. Okay, so how do we actually then see color? Well, the second possibility, if the trichromatic theory doesn't quite work for you, we have what we call the opponent process theory. And this is a different theory of color vision, which assumes that there's three opponent process cell systems, meaning that they're antagonistic, they're fighting one another. And you've got red and green, blue and yellow, and black and white. And this essentially says that the color information is processed after it's been processed by the cones. All right. So in other words, the actual processing of the colors is occurring deep inside the brain as opposed to on the retina at the level of the cones. And essentially how this works is that the colors in each system oppose one another, in that if one color is stimulated, the other one is inhibited. So if you stimulate the red, the green is kind of blocked. If you stimulate the yellow, the blue is kind of blocked. All right. And so that's how that the opponent process theory is thought to work. All right. Now, interestingly enough, it turns out that reality is probably not either trichromatic or opponent process. It's probably a combination of both. And we refer to this as the composite theory. We're taking both theories and kind of putting them together. And so the way this works, or the way
it's 9 o'clock.
Thank you, Jesus. The composite theory says that color information is processed by the cones according to the trichromatic theory, but it's then processed again after it's in the nervous system by the opponent process theory. So both theories are supported or encouraged by this composite theory. Okay. So we've spent a lot of time on vision because, again, it's arguably the most important sense that human beings use. However, the second most important sense that we have is our sense of sound or hearing. And so let's have a look at our ear, all right? This outside piece of the ear here, the flaps of your ear, are called the pinnae, and they work like catcher's mitts. All right, they essentially gather sound from outside our bodies, especially sound that's coming from in front of us. All right, if you ever see somebody who wants to hear something, they kind of go like this. All right, what are you really doing? You're increasing the size of that pinna. All right, and you're bouncing more sound waves down through the auditory canal. All right, and that's this tube that leads through the skull. All right, this is all bone tissue up here. All right, so this is your skull, and down here is your skull. And then this tube, the sound waves are coming in like so. Now, sound waves are actually transverse waves. I'm sorry, longitudinal waves, not transverse waves. But what happens is they hit here and they bounce off of the tympanic membrane, which is also known as your eardrum. The eardrum then begins to vibrate at the same frequency as the sound that you are perceiving. That moving of the tympanic membrane or the eardrum then causes three small bones, the malleus, the incus, and the stapes, these three bones here, to vibrate again at the same frequency. And what they do is the stapes starts banging on this organ right here. And this is called the cochlea. All right, now the cochlea is this long spiral-shaped organ. All right. And if you kind of stretched it out, it would look something like this. All right. And what happens here is that low frequency sounds stimulate hairs in the early portions here, and high frequency sounds stimulate little hairs over here. But the important aspect of this is that those are the sense organs, these little hair-like organs that are on the inside of this chamber. And when they get stimulated, they send signals down the length of the cochlear nerve through the rest of the skull and into the brain. All right, so if you're stimulating these hairs, you hear a high-pitched sound. You stimulate these hairs, you're hearing or sensing a low-pitched sound. The organs up here, these are the semicircular canals, and they help with rotational um, uh, equilibrium. All right, you know when your head is turning or moving. And then there's an area in here called the vestibule, which helps with gravitational equilibrium. In other words, you know which way is up and which way is down. Okay, so that's how the ear actually works. Now, I don't know why this is doing this. This is very strange. So let's talk about the cochlea, all right, that organ of hearing, okay? So what happens here to start is that there are the bones of the middle ear, called the stapes, vibrates and hits the membrane of what we call the oval window. It's just a little area of the cochlea that gets tapped by the stapes, and the frequency of the tapping causes fluid inside the cochlea to begin to form waves. Now, you ever drop a rock or a pebble or something in a puddle, right, or a pond, right? You note that the waves then spread out from wherever you dropped that object, right? Well, the same thing is going on here, all right? As the stapes taps on the oval window, it causes fluid waves. Now, those waves travel the length of the cochlea, and they cause little hairs on the inside to vibrate at the same frequency of those waves. The little hairs then send nerve impulses to the cochlear nerve, which eventually leads to the brain. Now, the pitch of sound is determined by varying wave frequencies that are detected by different parts inside the cochlea. All right, the organ of Corti, it's called. All right, high frequency waves give you a high frequency sound. Low frequency waves give you a low frequency sound. All right, so if you're talking like Elmo, that's a high pitched sound, right? And certain hairs are being stimulated. But if you're talking like Darth Vader, no, I am your father. All right, if you're doing that, then you're stimulating different hairs. And so different parts of the cochlear nerve are sending signals to the brain. Now, remember that the volume of the sound that you're hearing, the loudness of the sound that you're hearing, is determined by how much energy that sound wave has. So if you're in a library and you're whispering to the person sitting next to you and you're telling them all your favorite secrets, and there's hardly anything else that you might want to know, all right? Or if you're standing, underneath a fireworks display, boom, boom, the difference in the sound that you are perceiving in your brain is based on the energy of the sound wave that you hear. Okay, and so that's important in terms of that. Now, oh dear, I'm not really sure why this is doing this, but we'll just accept it for now. The hair cells inside the cochlea are the receptor cells. So just like the cones and the rods are the receptor cells in the eye, the hair cells inside the cochlea are the receptor cells for hearing. All right, and they line the inner membrane of the cochlea. All right, so We drew that in the slide a few moments ago. Now, um, issues with the ear include nerve deafness and conduction deafness. All right, difficulty in hearing. So nerve deafness is caused by damage to the hair cells inside the cochlea, or to the actual auditory nerve fibers inside the inner ear, from the cochlea into the brain. Um, Conduction deafness is hearing loss created by damage to one of the structures in the ear, one of the three bones, or the tympanic membrane, all right, or perhaps the oval window of the cochlea itself. All right, and so there's no bone to tap on, for example, on the oval window. All right, if the stapes is damaged, or even the malleus or the incus is damaged, you can't transmit the frequency of the sound into the brain. Okay, and so that obviously leads to deafness. All right, but deafness is caused there's either nerve deafness or conduction deafness, and they have to be treated very differently by a physician. All right, hearing aids are wonderful for certain things, like nerve deafness, um, or for conduction deafness, depending upon what kind of hearing uh, aid you actually are using. All right? And different ones need to be used for nerve deafness and conduction deafness. Okay, so we're going to do this again. All right, so the next thing that we're going to talk about is pitch, and this again deals with the frequency of the sound that we're dealing with, the Elmo voice versus the Darth Vader voice. Okay, and just like with sight and brightness, if you will, there are, and color theory, there are several theories about pitch as well, and how pitch is perceived by the brain. The first one is what we call the place theory. Okay, and this is a theory of pitch perception, which assumes that there is a specific location along the basilar membrane. All right, now remember, the basilar membrane is the inner membrane of the cochlea, that curly shaped organ. So if you stretch it out, all right, to its full length, what we're saying is, in the place theory, that there's a special spot in the cochlea where a certain frequency of sound is perceived, okay, or sensed, I should say. All right, so the place theory is a theory of pitch perception, which assumes that there is a specific location inside the cochlea or along the basilar membrane that maximally responds to a particular frequency. And then that particular group of hair cells sends a signal to the brain, and the brain says, "Oh yeah, that's Elmo," or "Oh yeah, that's Darth Vader." As this location goes down the basilar membrane from the oval window, the pitch goes down from 20,000 hertz. Hertz are just a unit of sound, or I'm sorry, a unit of frequency, um, down to 20 hertz. All right. And I think now that I'm looking at this, I think I said that backwards when I drew the picture of the cochlea and stretched it out. All right. The high frequency sounds are heard at, or perceived, sensed at the top of the cochlea, in the beginning of the cochlea, whereas the deepest, lowest pitched sounds are found way at the very tip top, the end of the cochlea. And if you think about that, that makes a lot of sense, because if you ever looked at, like, the cochlea, for example, of a whale, all right, it's just absolutely immense. All right, and so, because they perceive very low frequency sounds that can travel great distances through the ocean water. Now, the second theory here is what we call the frequency theory, and this is a theory of pitch perception that assumes that the frequency of sound wave is mimicked by the firing rate of the entire cochlea, all right, the entire basilar membrane. In other words, the hairs move at a certain thing, and based on the frequency that every hair is moving, the pitch is perceived. All right. Now, in reality, it's once again just like for vision, for hearing. There's also a composite theory. All right, and so where both the place theory and the frequency theory come into play here. And so, what this composite theory says is that low frequency sounds, sounds that have a frequency that is less than 500 hertz, all right, are processed according to the frequency theory. But very high frequency sounds like dog whistles and very, Elmo's voice, um, frequencies that are greater than 5,000 hertz. These are then processed according to the place theory. Okay, and so this is uh, again a composite of these. And of course, what that means is that we've got a whole range of sound between 500 and 5,000 hertz that are processed in both ways. That both the place theory and the frequency theory come into play here. All right. So lower frequencies are processed by frequency theory, higher frequencies are processed by place theory, and the frequencies in between are processed in both ways. Okay, so my friends, that brings us to the end of our discussion of the second section of, uh, of chapter 3. All right, this is the end of the second of 2 videos on how we see and hear. And I hope you learned something.